Hi, my name is Dominic Chang.
I'm Adya Kona.
I'm Sneha Anand.
I'm Lior Polishuk.
I'm Johnson Huang, and for our video, our group researched how gene editing and CRISPR are involved in cancer treatment.
Cancer is a battle that has been going on for a very long time and has been fought in a variety of different ways. The Mighty Chondrias present our research and presentation on one newer way that has a great deal of potential in the field, gene editing. Cancer, which is fundamentally a genetic disease, can originate from our DNA in many different ways. Tumor growth in and of itself is characterized by cells dividing and mutating in such a way that the processes of cell division and cell growth going forward in the body are negatively impacted. But on the path to getting there are a variety of reasons that this can actually happen. In addition to chance mutations born of pathogenic and/or carcinogenic exposure, which are called acquired mutations, there are also genetic variations that we can inherit, either from our parents' genomes or from mutations that occur in gametes during fertilization. The conditions caused by these mutations, which scientists have labeled as hereditary cancer syndromes, can promote the likelihood of cancer development in various ways and predispose people to a higher risk of getting cancer in their lifetime. There are three main types of genes that, when mutated in some way, typically lead to these conditions. There are oncogenes, which are altered versions of the proto-oncogenes that typically promote cell growth. There are tumor suppressor genes, which naturally produce proteins to inhibit tumor formation. And then there are DNA repair genes, which are involved in the complex processes of genomic and cellular replication. Specifically, the most common gene that appears to be mutated in cases of cancer, at least as scientists have observed, is the TP53 gene, which normally produces a protein that suppresses the growth and development of tumors, a tumor suppressor gene. Inherited mutations in this gene can lead to a rare autosomally dominant syndrome known as the Li-Fraumeni syndrome, which lead to a higher risk of developing a large number of cancers, such as breast cancer, osteosarcoma, and neoplasms of the head, neck, and lungs. There are numerous other examples, too, from the impact of the BRCA1 and 2 genes on hereditary predisposition to breast and ovarian cancers, to the role that mutations in PTEN, another tumor suppressor gene, play in the inherited Cowden syndrome. And there are many, many more. But now that we've charted the very tip of this iceberg, that is the leagues of ways in which cancer can be caused by genetic factors and variants, what sorts of solutions to these ailments have modern science as we know it, or maybe don't quite know it yet, come up with? Meet CRISPR.
So what is CRISPR? CRISPR is a simple tool for altering DNA sequences and modifying gene function to correct genetic defects and treat or prevent diseases. CRISPR stands for clusters of regularly interspaced short palindromic repeats. It is based on natural defense mechanisms of bacteria and archaea, which use RNA and various CAS proteins to foil attacks by viruses and other foreign bodies. The CRISPR region of DNA is a specialized region with two distinct characteristics: nucleotide repeats and spacers. Repeats are building blocks of DNA distributed throughout the CRISPR region, while spacers are bits of DNA that are interspersed among these repeated sequences. In the case of bacteria, the spacers are taken from viruses that previously attacked the organism. They serve as a bank of memories, which enables bacteria to recognize the virus and fight off future attacks. According to a 2000 paper by Rodolphe Barangu, when a virus attacked Streptococcus thermophilus bacteria, commonly found in yogurt and other dairy cultures, new spacers were incorporated into the CRISPR region. The DNA sequence of the spacers were identical to parts of the virus genome. Once the spacer is added and the virus attacks again, a portion of the CRISPR is transcribed and processed into CRISPR RNA, or cRNA. Each cRNA consists of a repeat and a spacer, according to Jennifer Doudna and Emmanuel Charpentier in their 2014 paper. The Cas9 protein, an enzyme that cleaves DNA to switch off a targeted gene, typically binds to two RNA molecules, cRNA and another called transactivating cRNA. These two RNA strands are what make up the guide RNA, or gRNA. Like how it is named, guide RNA guides Cas9 to the target site on a double-stranded DNA, where it will make its cut. Using two separate domains on its structure, Cas9 cuts both strands of the DNA. This cut then shuts the target gene off. Scientists have modified Cas9 to activate or repress specific genes as well. By modifying two amino acids in Cas9, the enzyme becomes a mutant that is not capable of cutting DNA, but instead can be programmed to either activate or repress gene expression at that position in the genome. This protein is called nuclease deficient endonucleases. To make sure that Cas9 does not just cut anywhere in the genome, short DNA sequences known as PAMs, protospacer adjacent motifs, serve as tags and sit adjacent to the target DNA sequence. If the Cas9 doesn't see a PAM, it won't cut. CRISPR in computational biological terms. Step 1. Decide your target gene. Modification of the specific gene can either be to cut, activate, or inhibit its gene expression. Step 2. Choose an endonuclease protein. Cas9 is the most commonly used protein for genome engineering, but there are other proteins such as CPF1. Step 3. Design a gRNA of 20 nucleotides to guide Cas9 to target genes. Crossing a gRNA with a high on-target score indicates that the gRNA will bind very well near a particular PM sequence. Choosing a low off-target score suggests that there are DNA sequences similar to the target gene elsewhere in the genome, which can be confusing for the gRNA about where exactly to bind, resulting in decreased efficiency. So it's important to choose a gRNA with high on-target and off-target scores. Step 4. Create an expression vector to express both Cas9 and gRNA. An expression vector, or bits of plasmid that actually make up the Cas9 gene enzyme, can be isolated from living bacteria after they have been destroyed. After that, you can engineer the cells and wait for colonies to appear. These new offsprings will have the modified version of the gene.
There are many different ways CRISPR has been applied to cancer research, and here are just a couple of them. Giving T cells a protein receptor that can discern NY-ESO-1, modifying T cells to show certain receptor proteins, assisting with breast cancer research, and helping with metastasis studies. One of the methods for CRISPR, whose first human trial was at UPenn in 2019, involved using CRISPR to modify the T cells, which are a major part of the immune system, and making them better detect and fight cancer cells. The first step in this process is adding a synthetic gene that gives T cells a protein receptor that allows them to recognize a molecule named NY-ESO-1, which has showed up in many cancer cells, including myeloma, metastatic melanoma, bladder cancer, prostate cancer, and breast cancer. Next, CRISPR is then used to remove two genes that interfere with the NY-ESO receptor and a gene that interferes with killing cancers. A study tested this method on 3 patients, 2 with advanced multiple myeloma and 1 with metastatic sarcoma, all which had NY-ESO-1. The results showed this method to be a safe treatment, though only 10% of T cells had all of the gene edits. There were also off-target edits, but unfortunately they were not harmful. However, the CRISPR gene editing only halted the growing cancer for a short while for 2/3 of the patients, but it is promising as a treatment for solid tumors. To sum it up, some drawbacks are that the treatment's long-term effects are not totally clear and that monitoring and future improvements will still be required. However, there have also been other studies using CRISPR for CAR T-cell therapies to test for B-cell and myeloma cancers. So this CAR T-cell method was first tested in mice and works by modifying cells to make them show CARs, or chimeric antigen receptor proteins, which helps them detect and attack cancer cells with the same antigen. The antigen is CD19, which is known as a biomarker for B cells. The CD19 antigen is shown in very high quantities or low quantities when there are B cell malignancies, which can help detect lymphomas and other cancers relating to B cells. The CAR T cell therapy entails returning a patient's T cells after they are genetically modified using CRISPR to improve their attack towards tumor cells. The old method was to use a retrovirus to deliver the CAR gene, but it inserted randomly, so it was inefficient and likely dangerous. The CRISPR method, however, allows researchers to insert the CAR gene in a special area called the T cell receptor alpha chain genome. The CAR gene is specifically inserted in the CCR5, short for chemokine, CC motif receptor 5, and AAVS1, known as adeno-associated virus site 1 loci, which are two of three relatively safe genomic harbors. When they do this, they insert it into the T cell receptor gene after deleting a certain part. These two methods were tested on mice with leukemia, and the CRISPR technique proved to be much more effective, along with preventing the likely scenario of CAR T cells beginning to not recognize and fight tumors over time, also known as exhaustion. CRISPR has also been applied to many different aspects of breast cancer research. For example, it has been used to discover that the cyclin-dependent kinase inhibitor 2A, CDKN2A, is the cause for rapid cell division, that targeting the HER2 gene helps inhibit tumor growth, and lastly, that the BRCA1 delta 11q alternative splice isoform actually forms resistance to breast cancer therapy. Lastly, CRISPR is also used to study metastasis, which is when cancer spreads to different areas of the body, by using Cas9, likened to Pac-Man, to make small cuts every time a cancer cell metastasized. These edits were then used to help researchers determine the frequency of the cell divisions and where the cells changed.
Next topic will be on how CRISPR compares to current gene editing methods. ZNFs were the first of genome editing nucleases available, and they are also the most common DNA binding domain found in eukaryotes. They typically are comprised of around 30 amino acid modules that interact with nucleotide triplets. ZNFs have been designed to recognize all of the 64 possible trinucleotide combinations, and by stringing different zinc finger moieties, we can create ZNFs that specifically recognize any sequence of the DNA triplets. Each ZNF typically recognizes 3 to 6 nucleotide triplets, and because the nucleus to which they are attached only function as dimers, pairs of ZNFs are required to target any specific locus, one that recognizes the sequence upstream and the other that recognizes the sequence downstream of the site to be modified. The next method is TALENs. TALENs are similar to ZNFs in that it also uses DNA binding motifs to direct the same non-specific nucleus to cleave the genome at a specific site. But instead of recognizing DNA triplets, each domain recognizes a single nucleotide. The interaction between the TALEN-derived DNA bind domains and their target nucleotides are less complex than those between ZNFs and their target trinucleotides, and designing TALENs is generally more straightforward than ZNFs. A key advantage of both techniques is that they are not limited to mutagenesis in mouse embryonic stem cells. ZNFs and TALENs modifications have been engineered in zebrafish, fruit flies, rats, livestock, and even in monarch butterflies. The latest development in genome editing is CRISPR. CRISPR systems are RNA-based bacterial defense mechanisms designed to recognize and eliminate foreign DNA from invading bacteriophages and plasmids. They consist of a Cas endonuclease that is directed to cleave a target sequence by a guide RNA. Both the Cas endonuclease and the RNA are encoded by an array in the bacterial genome, and the system can be co-opted to cleave any target sequence of choice by modifying the sequence of the RNA.
Although CRISPR is a very recent method of medical research and treatment, it holds bright promise for future cancer treatment. The first clinical trial using CRISPR to treat cancer, started by the University of Pennsylvania, is still ongoing. However, it has concluded that CRISPR is somewhat effective in treating different types of cancer, especially solid tumors, but does not fully stop tumor growth. Another previous study published by the American Society for Clinical Investigation involved misguided cuts, which resulted in treated individuals developing T-cell leukemia in a range of 31 to 68 months after the gene therapy was applied. Essentially, more cells became cancerous rather than less. In addition to these trials, research published in the Oncology Times claims that the most difficult challenges for CRISPR treatment include safe and efficient delivery to the specified cells, since existing delivery systems can potentially be toxic or damaging. Another challenge mentioned was unintentional off-target editing, such as the misguided cuts previously mentioned. Even if CRISPR gene therapy to treat cancer was perfected, it would likely cost millions of dollars at first as well. These trials and research have made clear the primary concerns about current CRISPR treatment, including safe and efficient delivery to the specified cells, unintentional off-target editing, and expensive costs. If these concerns are addressed in the future, many scientists are confident that CRISPR can become a viable and effective treatment for cancers.
These are our works cited. As you can tell, we did a lot of reading for this. Thanks for
Thanks for
watching.
watching.
Thanks for watching.